So, good morning to everyone and good evening to Professor Elizabeth. She's here. So, before I start, I request Dr. Amit Sharma, Director at National Institute of Malaria Research, that he'll open the talk and later I'll talk about, I'll read about a few lines about Professor Elizabeth and his work and her work. So, over to you, sir. You can start now.
Yeah, well, good evening, Dr. Winzeler, and good morning to everybody. It's a real special privilege for me as well personally and for NIMR and for MERA-India to host Dr. Winzeler's talk today. Many of you, or almost all of you, should have been reading our papers, but many of you have been reading about drug discovery work and some of the really astounding scientific screen work that Dr. Winzeler has championed in malaria, and phenotypic screening is, of course, critical component of drug discovery for malaria. And I'm sure you will enjoy the journey today of some of the best science that is possible in this field. And I'm really grateful to Dr. Winzeler for accepting a talk time, which is 9:30 her time, I take it. So it'll be running late for her. But so she's sort of, I hope she's had a dinner, etc. We'll try to keep the questions to a minimum so that she can go to bed early. But thank you very much, Dr. Winzeler, for this. And we really hope in future that you will be able to visit NIMR and also its field stations. We have 8 field stations across India, and that maybe potentially we could collaborate. So thank you. And we are really excited about your talk.
Okay, thank you so much. Yeah, so before we start, I'll take a few minutes here. So this is an unplanned lecture series which we were, Dr. NMR and MERA-India, we were trying to get big shots in the field here, and luckily, fortunately, we have now the calendar is full with big shots like Professor Elizabeth. So after Professor David Stewart's talk, who talked about the changing antigen and anatomy of SARS-CoV-2 spike, I'm really glad that today we have Professor Elizabeth with us, where she will talk about Malaria Drug Accelerator. We are very grateful that she accepted our invitation. I know there's a lots of back and forth on emails, and finally we decided that 24th is the day, and she agreed to talk despite her busy schedule, and she is an early bird, but she agreed to to talk about her science and her work late night today. Dr. Elizabeth Winzeler is a professor at University of California, San Diego School of Medicine. She obtained her PhD and performed postdoctoral work at Stanford, and before moving to a joint position at Scripps of Novartis Research Foundation in 1999. At the Genomic Institute of Novartis Research Foundation, she led a malaria drug discovery program that discovered several novel antimalarial chemotypes that have progressed into clinical trials. In 2012, she moved to the University of California, San Diego, where she leads a group that uses systemic data-intensive methods to solve problems at interface of post-pathogen biology typically involve large collection of chemical screening data and whole genomic sequencing. She is currently the director of Malaria Drug Accelerator program. She has received awards from Keck Foundation, the Ellison Medicine Foundation, and Bill Melinda Gates Foundation. In 2014, she was awarded the Bailey Ashford Medal for Distinguished Achievement in Tropical Medicine. She has authored more than 200 scientific publications, and she is a very, very well-known name in malaria research. The Malaria Drug Accelerator is a consortium of 18 laboratories whose mission is to discover both high-quality targets for antimalarial drug discovery, develop high-throughput assays, and discover easy leads. And where she will in her lecture she will describe their approach to finding high-value targets for antimalarial drug discovery. So I would now, before over to Professor Elizabeth, and you can ask your question and answer in a chat. You can type your questions in your chat box, and we'll try to cover as maximum as possible to answer. Okay, so with this, I would like to invite Professor Elizabeth. Professor Elizabeth, over to you.
Okay. Thank you. And can everyone hear me? Yeah.
Yes.
Yeah, I decided to, I'm going to be talking about the Malaria Drug Accelerator, but I thought it would be fun to talk a little bit about how we got here and, you know, what we've been doing for the last decade a little bit. So I'm going to give a bit more history and put some of the articles that we've published into context. So a little bit more about my journey. I'm sure everyone in this audience knows the background of malaria. And most people, I'm sure, know that there's no vaccine for malaria, that malaria is primarily prevented by using insecticide-treated bed nets and spraying with insecticides. And once you get malaria, it's you know, primarily treated with drugs, and there are effective drugs, but drug resistance is one of the big issues. And you may know also that after years of decline, malaria is starting to come back again. So there was a big surge in malaria about 20 years ago with chloroquine resistance emerging, and then there was a lot of investments in malaria and malaria drug development, but also malaria control. And a lot of this was, you know, distributing insecticide-treated bed nets, distributing medicines. And there was a big decline in malaria, and people were thinking that maybe we could start thinking about eliminating it 100%. And including eliminating it from Africa. But over the last couple of years, you know, the number of cases has stopped dropping, and has even started to inch up again. And a variety of different countries are now reporting increases in the number of malaria cases, and it's suspected that with the COVID-19 situation, it will be even worse in the 2020 World Malaria Report. And as you also probably know, treatment failures are now becoming fairly common in Southeast Asia, in particular in Thailand and Cambodia and Myanmar. Parasites are resistant not only to artemisinin, which is the primary drug used to treat malaria, but also to the partner drugs. It is possible that you can contract malaria in some of these areas and have no treatments available, which can ultimately, you know, lead to a big increase in fatalities. So that's kind of scary. One of the problems with malaria, though, is that, you know, by the time you're actually getting a treatment, you've already got the disease. And what we'd really like to be able to do is to prevent malaria. And because if you can prevent malaria, you're probably going to prevent most of the deaths. If you don't get malaria, many people die, especially in sub-Saharan Africa. The children often die from malaria just because you know, they don't get to the hospital in time. And a lot of the decreases in the number of cases that people have been seeing. I'm going to move this. Sorry. Got this little picture right in the middle of my slides. Okay. Another reason that malaria cases may be going up, it may be drug resistance, but another issue is insecticide resistance. And a lot of malaria cases the number of cases that have been driven down has been through the use of insecticide-treated bed nets. And a lot of the mosquitoes that people are sampling out of, especially in sub-Saharan Africa, are now resistant to most of the different insecticides that are currently in use. And that's also creating an increase in the number of malaria cases. So we, to address the drug resistance issue, we need to develop new treatments for malaria. But I would also argue that the current drugs may not be the best choice. So even though the development of artemisinin combination therapies has had a huge impact on people's lives, it has issues as a drug. I mean, this is a natural product derived drug. It's derived from a tea that was known to have antimalarial properties 2,000 years ago. And the thing about it is that it does make you feel better, but it only makes you feel better after you're sick. And it certainly doesn't prevent you from getting malaria. And chloroquine, which is derived ultimately from quinine, and it was also a natural product, also makes you feel better, but it also doesn't prevent malaria. And the worst thing in both of the cases of these two drugs, neither of these are particularly active against the gametocytes. So I don't know if we have students in the audience, but the malaria life cycle, which is shown right here, we all know that malaria is transmitted by mosquitoes, and when the mosquitoes bite you, they release sporozoites in your bloodstream and they travel to your liver. And we only get symptoms of malaria once the parasites emerge from the liver and start replicating in your in your blood cells. And that's when you get the cycles of fever and chills, the anemia, the cerebral malaria. And when the parasites are stressed, they will differentiate into male and female gametocytes. And these are the only forms of the parasites that are responsible for transmission. And chloroquine and artemisinin, neither of them have much impact on gametocyte carriage or are able to kill gametocytes. So you can take both of these drugs, and what happens is that you'll feel better. Your life will probably be saved, and that's great. But you can continue, in some cases, to transmit malaria. And so what we would like, in addition to drugs that overcome the resistance problem, is to thinking about creating next generation drugs that have properties that are missing from the current set of drugs. And so we'd really like to have drugs that are more potent than the current drugs. We'd like to have ones that don't have resistance liabilities. We'd like compounds that block you from getting malaria in the first place. And we would like to have compounds that ultimately block malaria transmission. And so, the thing that I've been thinking about really for the last 15 years is how can we, step outside and really try to think about creating completely new sets of compounds that are better than the old compounds that are currently in use. So my journey on this started about 15 years ago when we decided to see, at the time, most people who were doing antimalarial drug development were doing what I call Me Too chemistry. So they would take an aminoquinoline and they would add a new halogen to it and try to improve its PK. But they were working largely on the same sets of scaffolds that people had worked on before. And making new versions of artemisinin, and we just had this idea, which was, well, maybe we should just try to toss a whole bunch of different compounds on malaria parasites and try to figure out what kills them, and then see if we can make drugs out of these compounds. And the approach is kind of a traditional one, but what we had at the time was access to a lot of robots and liquid handling machines, instruments that allowed us to go through and test many compounds in very, very small volumes. So we were able to test compounds' ability to kill malaria parasites in 1536 well plates. And this allowed us to go through very, very large numbers of compounds. And we ran a screen in about 6 weeks where we basically just incubated nanoliter, like 10 nanoliters of parasites with, a probably picoliter volume of compound, and it's very hard to dispense picoliters, or well, nanoliters. I think 50 nanoliters, I think, that went into this. It's very hard. You need a machine to do it. Okay, so this is the trick. It's not that it was conceptually that difficult. It was just that developing a way to dispense these tiny amounts of compounds and get really reproducible readouts was the challenge. But we did it, and this was in about 2005 and 2006, and we, to our delight, we discovered that there were many compounds in this libraries that we looked at that looked nothing like things people had identified as antimalarials in the past. So we knew it was working because we found many of the existing antimalarial compounds, but we found all sorts of new things too. And this was really one of the very first large-scale, high-throughput screens that had been run against malaria. And, we took some risks in terms of finding compounds and then trying to see if we could turn them into new drugs. So the first compound that I was associated with was K609. And this came actually from kind of a pilot phenotypic screen that we ran when I was working at GNF. And the first compound that we identified, we had about a 200 nanomolar, 200 nanomolar, and through a few modifications, we were able to turn this into something that was even more potent. And we were able, without too many modifications, to improve the PK. And we were able to then test it in animal models of disease, and to our delight, again, it was able to cure mice, and to cure mice more rapidly than the gold standard treatments. So that actually progressed into human clinical trials very quickly with my colleagues at Novartis. And this is still now, it's continued to be safe, it's continued to be efficacious, it is able to cure patients. Unfortunately, this says something about the slow pace of drug development. We moved very quickly in the first couple of years to get proof of concept in man, but the subsequent years have not been kind. It takes a lot of resources to do clinical trials. We're still only in phase 2B. This is being looked at for treatment of severe malaria. And it's continuing along, and hopefully this will be licensed soon. But that was one of the first things that we came up with. The next thing that we did, and this was running kind of in parallel, was we were intrigued with this idea. Well, K609 can rapidly relieve the symptoms of malaria, but can we make even better drugs? And for this, we did a cross screening where we took compounds that had blood stage activity that came from this first big screen, and then we subsequently tested them for their ability to block malaria parasites development in liver stages. And we developed a semi-high throughput assay to monitor parasites development in liver stage. And for this, what we did is we had to get malaria infected mosquitoes. They were infected with P. burgii, a rodent malaria parasite. And we had to dissect out the sporozoites from these mosquitoes that had been fed on mice that were infected with malaria. And then we would put them on lawns of hepatocytes or HepG2 cells. And you could monitor their development using automated microscopy and an antibody to HSP70, parasite HSP70. And you can see some of the parasite schizonts developing here. And so we took many of the compounds that had activity in the blood stage assay, and then we retested them against liver stage parasites, and then identified compounds that not only were able to kill these asexual blood stage parasites, but also prevented malaria by acting against liver stage parasites. And through that work, we found a second compound that was optimized with colleagues such as Adi Nagel and Arnab Chatterjee, and we took kind of a cheminformatics approach where we looked at lots of different compounds and looked for compound classes that were enriched in our screening hits. And we decided on this imidazole piperazine scaffold family, which we found 3 different examples, all of which were fairly potent through clustering, and decided to make modifications to this. And this resulted in a compound called CAF156, which is also in phase 2B clinical trials. And again, we were just modifying around the scaffold. We didn't know where it was acting, but ultimately created something that was fairly safe and efficacious first in animal models. And this is then was tested for toxicity and then progressed to human trials. And this also showed very nice activity in human malaria patients. Enabled and this compound CAF156 is now being paired with lumefantrine. It's being developed by Novartis, and it's also in fairly widespread human trials. And I would also like to, I'm kind of pleased that our compounds have done well, but the general approach of taking compounds that are active in these phenotypic screens and then developing them was an approach that was kind of picked up by a variety of other people, and not only our compounds, but compounds from the Kip Guy's lab and from other investigators are also making their way into human trials, and hopefully some of these will be licensed in the next few years. More recently, we've been trying to think about, can we think about finding molecules that can prevent malaria? And can we make thinking about long-acting chemoprophylaxis? I mean, the idea here was, it's a real pain to go around and try to stay under a bed net all the time. And what if you could prevent malaria, by taking a shot and prevent malaria for 3 months? And if you could be protected from malaria for 3 months, that might be enough to kind of break the cycle of transmission. And so we did another screen that was published a few years ago where we started with just started with liver stage parasites. Here we had to really drive down the cost. It's pretty expensive to grow malaria parasites and to dissect them out of mosquitoes. It's still no machine for dissecting mosquitoes. And you basically have to have undergraduates or various helpers, putting the mosquitoes out and pulling out their salivary glands with needles. And so we needed to make a really sensitive assay in order to go through a large number of compounds. And we used a luciferase expressing parasite here. And this is just an example of one of our screening plates where if the parasites are happy, they let out a lot of light signal. And in the cases where you have a dark spot on this 1536 well plate, it's a place where a compound has killed the malaria parasites and then prevented them from growing. And we then went through about 500,000 compounds and identified a lot of different things that are possibly acting that really just prevent malaria. Some of them act against the blood stage, but some of them just simply act against the liver stage. And I don't have time to talk about it, but I think there's some really exciting preliminary data suggesting that there's a lot of interplay between parasites and hepatocytes, and it's much more complicated in the liver stage. And there may be opportunities to disrupt that host pathogen interface and possibly block malaria development. So this is newer. I can't say there's any compounds from this activity that have progressed to clinical trials at this point, but hopefully in a few years we'll see something coming out of it. This is a picture of my laboratory at UCSD, and these are some of our undergraduate students who are dissecting mosquitoes here. And that's how we were able to kind of run the screen. And we had to run it over about a year and a half because of the availability of mosquitoes. And, basically this worked pretty well. One of the things that we did is the same thing we did with the CAF156, which is to do compound scaffold clustering and to show that you get enrichment of particular families. And this gives you an idea that things are working. And we found, a bunch of compound families that we knew would be have antimalarial activity, and we found them at rates much higher than what you expect by chance. For example, this is a scaffold family that we know that now hits PI4K. If you do clustering of these compounds on 500,000 that were in this library, we found 17 family members in the entire library, and 16 of the 17 family members were active against the liver stage parasite. And the probability of having that by chance is 1 × 10⁻15. So without even doing much additional work, you can, through the use of statistics, you can often get a good idea of what, what compounds are interesting. So again, I'm proud of the fact that many of the new drugs in the antimalarial drug development pipeline are now coming from phenotypic screens, and it's not just ours. So, I talked about cypargamin, I talked about CAF156 that came from screens that I ran, but several other ones like MMV0048, that's a PI4K inhibitor, SJ733. So I want to get into talking about the Malaria Drug Accelerator for the last little bit. So the problem with doing phenotypic screens and to just going through lots of compounds is that it's actually very costly. And I alluded to the fact that it costs a lot of money at several occasions. It's not too bad for a blood stage screen where you're just incubating minute amounts of parasites with blood cells and compounds. But if you're looking for compounds that are active in the liver stage or worse in a radical cure assay, where you actually have to infect cynomolgus monkeys, it becomes very, very expensive. And it can only be carried out in specialized laboratories, malaria labs. That means that you're limiting the amount of talented people that can contribute. And if you do a phenotypic screen and you get a compound out, you may not understand if it fails, where that toxicity is coming from, because you can't counter screen it against a human target and or optimize it based on selectivity for the parasite target versus the human one. And this limits the fact that the screens are expensive limits the number of molecules that can be screened. And so theoretically, a better approach is to use target-based drug discovery and to use crystal structures to try to specifically design compounds that inhibit a particular target. And in theory, this can give you more potent compounds, ones that might not need to be given less, and it allows drug discovery to be done in more places as well. So how do you find the targets of bioactive compounds? So this is something I've been working on with the genetics background for many years. And it started back in the days of K609. And we thought what we could do was to possibly find the targets of these compounds by raising drug-resistant parasites. We didn't know if this would work. It was actually pretty exciting work when we showed that it did work. But the idea was that you would take parasites and clone them by limited dilution because you wanted to do good microbiology. And then after you clone them by limiting dilution, you would apply a sublethal concentration of the drug that you're interested in while maintaining parasite growth at a fair rate. So we try to keep like 10 to the 8th parasites in the flask at all time, and to keep them so that they're still growing, but not growing as fast as when the drug is not present. So they're just kind of percolating along. And then eventually you can ramp up the drug a little bit, and then parasites will eventually acquire a beneficial mutation that will allow them to start growing faster. And when you see this, you can then clone your parasites again and then try to figure out the mutation that's causing the increased growth. And initially, before we had whole genome sequences, we used to use microarrays to try to compare the genomes of the parasites, and I was involved in pioneering that. But now we tend to do whole genome sequencing. And you kind of need to develop some specialized pipelines, but by doing this multiple times, we kind of got pretty good at it, and filtering out the mutations that are likely to be unimportant, and these are often mutations in multi-gene families. The multi-gene families seem to mutate at higher rates. But if you focus on, the core genome, typically you only get a few mutations in the entire 25 million base pair genome, and it allows you in some cases to pick out the drug target. And this is just an example from phenylalanine tRNA synthetase, a paper that was published with Stuart Schreiber in the Worth Lab a few years ago. And these are the sorts of mutations. And we discovered that this compound, we had no idea what it did, BRD1095, is probably hitting phenylalanine tRNA ligase by virtue of the fact that we found 4 mutations in phenylalanine tRNA ligase in 3 independent clones. And the probability of finding 4 mutations all in the same gene when you have 6,000 genes in the genome and you have 3 independent clones is, again, something times 1 times 10 to the minus 15 or something. It just doesn't happen by chance. So that gives you a very good idea that it's worth investing in phenylalanine tRNA ligase. And in the best situations, the mutations will often explain something about how a compound interacts with the active site. And in this case, a lot of the mutations that we identified map to a specific area that allows you to make hypotheses about how the compound is acting. So we've been recently, and this is the last couple of years, trying to exploit this with the Malaria Drug Accelerator. And the question is, how many targets can you come up with? And this is kind of difficult work. So what we've done is we have now a consortium that's funded by the Bill and Melinda Gates Foundation, where some of us will do resistance selections. We do a lot of phenotypic screening, and then we do this whole process in mass and try to find as many different targets as possible. And then we work to try to develop an assay around the target and try to lead this into lead optimization programs. And this slide is unfortunately probably a little bit out of date. We now have 18 members, but this is a group. This is mostly my core collaborators: David Fiddock, Dan Goldberg, Diane Worth, people I've been working with for over 10 years, as well as a variety of different industrial partners. And the objective is to, keep people from working on the same compound in different laboratories in different countries, and to try to share everything that we're doing, and to function a little bit like a virtual pharmaceutical company, where we send things around the globe. So David Fiddock may do the selection. My laboratory does the whole genome sequencing. We identify mutation in my laboratory. We work with Marcus Lee at the Sanger Center, who specializes in doing CRISPR validation. And then we send the compound over to Jacqueline Niles at MIT, who has a conditional knockdown system. And then maybe we'll work with a crystallographer to try to get a structure and to get an assay development going. And ultimately, this can go much, much faster than if you tried to work on all of this in your own little laboratory. Phenylalanine tRNA synthetase is an example of a target that has come through that has used MalDA resources, and this is now yielding new molecules that have been discovered by using structure-guided drug discovery. And there's a variety of other examples that we have. We've actually hit a lot of tRNA synthetases through this work, and that's partly because a lot of our compounds we're interested in are also active in liver, and tRNA synthetases are important for liver stage development as well as blood stage development. And you've probably seen some of the publications. Cyclin-like kinase 3 was another target where we had a lot of MalDA resources and used the same approach. This was a project that was led by Andrew Tobin, and we helped out in terms of putting things forward. And so I don't want to give the idea that this is very exclusive. We work with all sorts of people. We have the MalDA core investigators, most of whom have Gates funding, but people come and go. And so, for example, if you're interested, if you have drug-resistant parasites and you want to have a specialist sequence it for you, please feel free to reach out to me. We can sequence things really quickly and then compare your data to all of the data that we've assembled. That was the publication for that. And so these are just some of the various targets that are now being evaluated by MalDA. They've resulted in a lot of nice publications. And for example, isoleucyl tRNA synthetase, we're trying to get a high throughput assay going. And ultimately, we're really interested in creating better drugs. So that's a little bit of what we've been doing. It's been very collaborative. I think if you'll see any of our publications, you'll see that, we often have 15 or 20 different authors. And, it's difficult to work in malaria, but by sharing and being completely open about what you're doing, it can often go faster. And I think the funders recognize that this is a successful way to do things. People often ask, what happens? Aren't many of these targets are also found in humans. Again, you can engineer in specificity during drug development, and I don't have time to talk about it, but sometimes we use model systems like Saccharomyces cerevisiae to try to find targets. We've done this with other apicomplexan parasites and with fungi, and we've done a little bit in cancer. And the mutations that we discovered using in vitro evolution are often clinically relevant. They're actually often the same mutations that you'll find in field isolates. But we also need to be thinking about how to resist resistance and how to go about finding compounds where you don't have a resistance liability. I think that's something that we're spending a lot of time thinking about. And we're still finding new chemically validated drug targets, though I think we're going to be switching the field a little bit more into other approaches over the next couple of years. And so this is my lab, a bit out of date. It's actually not nearly as big as this. We have a lot of undergraduates that are here. I have about 10 people in my lab usually. But the most important group is all of the MalDA collaborators, some of whom are mentioned here, in particular Diane Wirth, David Fiddock, Dan Goldberg, and many other participants. So sorry for going a bit long, but I'll take questions if you have them.
Thank you very much. So, okay, so as we discussed, we have some questions here. Can you see the questions, Professor Elizabeth?
I can't see the questions because I'm.
Okay, I'll leave. Okay, question is, which compound did you find more promising in clearing parasite? Did the compound show any side effects?
Well, the ones that are in clinical trials really have very few side effects because it's very hard to find to make drugs that have to advance them if they have side effects. Maybe in the old days, you could develop drugs that gave you side effects, but now we really try not to have them. But I don't know. There haven't been many side effects for either CAF156 or K609 that I know of, but they haven't been tested in really large groups of people. And in many cases, the side effects only show up once you've given it to a lot of people. There might be people that have particular genetic alleles that make them react with a compound, for example. So at this point, I don't know what the side effects are going to be, if there are any.
Okay.
We'll see. I mean, I know the current drugs have side effects. I mean, I think mefloquine is well known for giving you nightmares, right? So.
Have you tried the effective compounds in combination with artemisinin? If yes, have find any combination of compounds effective against artemisinin resistant strains?
Yeah. So I wasn't involved in pairing. So CAF156 is being paired with lumefantrine, which is the partner drug for coartem. There was a decision not to use artemisinin as a partner drug because of the resistance that's known in Southeast Asia. So I think in general, people are trying to go away from using artemisinin type molecules in partners, as partners for some of these new molecules that have come from phenotypic screens. And this isn't just the work that I did that is now being developed by Novartis. There's also compounds that are coming out from other groups, and to my knowledge, they're generally not being paired with artemisinin, though in all cases, when they go to the field, they will have a partner drug.
In addition to mutations in the drug targets, what are the probable effects of mutations introduced by the compounds in other genes vis-a-vis effective on drug efficacy?
That's a very good question. It's something I didn't really have time to go into. So in most cases, we don't think the compounds are making the mutations, for one thing. We think that the mutations come from just random DNA replication, error-prone, just errors that occur. And, we can do calculations, and basically you just have to grow the parasites a very long time in the presence of your compound, and then the mutations will randomly happen, and then we select them out. But yes, there are other types of mutations that we get. So we can take all of the data that we've generated, and we've sequenced and analyzed about 500 different parasite clones at this point, and they're all isogenic. And then we can look through this list and find all the genes that we see over and over again. So we get targets, that's great, but we've also found a whole bunch of different transporters that are repeatedly mutated. The most commonly mutated transporter is PFMDR1. That is a real liability problem with a lot of compounds. They have MDR1 resistance, and that comes up as copy number variants as well as point mutations in PFMDR1. But there's a bunch of other ones too, such as the amino acid transporter 1, PFCRT, cyclic amine resistance locus, PF-CARL, and in many cases we've hit them many times, maybe 10 times, and these are at rates way more frequent than what you'd expect by chance. And most of these mutations in these transporters that we're seeing can be readily validated. So we have interests in studying drug resistance. We're working on them a little bit, but they're a little bit peripheral to the goal of trying to find new drugs and new drug targets. But I could give a whole other talk on transporter mutations.
Okay. If you permit, may I take one more question here?
Yeah.
So, is drug resistance always associated with genetic mutations in strains across geographical location, or in some cases, only differential expression of genes pathways are observed?
Yeah, I have studied this in a fair amount of detail. To be honest, I would say in almost all cases that I've looked at, it is a genetic mutation that confers drug resistance. And in the cases where there's some things that reported that maybe it was an epigenetic mechanism or a parasite I think a little bit deeper sequencing ultimately showed that there was actually a copy number variant that was not detected, and that contributed to the drug resistance. So I would say in most cases there's a mutation, and if you can't find it, you haven't looked hard enough. And I would say with almost all of the resistant clones, at least 95% of them, we get either a highly represented, we find a target with mutations in it at a rate not expected by chance, or we find one of our other culprits. such as PFMDR1, PFCRT, or other multi-drug resistance genes that appear repeatedly. So in terms of transcription, we seldom see in malaria any mutations in transcription factors associated with drug resistance. We see mutations in AP2G sometimes, and that's the transcription factor that's associated with commitment to gametocytogenesis. But we believe that these come up because we culture our parasites for a long time, and when you keep parasites in long-term culture, they often develop mutations in genes associated with gametocytogenesis. Now, that's just malaria, because we've done a whole bunch of similar studies using Saccharomyces, and in fungus and Saccharomyces, we see mutations in transcription factors all the time. I mean, that is really driving the drug resistance response in Saccharomyces. So we just think that they're different organisms. We don't think, at this point, that transcriptional mechanisms are that important for drug resistance. But that's only true for this particular species in Saccharomyces and Candida albigans and Aspergillus. Transcription factors do drive a lot of the drug resistance programs.
Okay, so I'll take one or two more. This is, what's your take on in vitro evaluation against these new antimalarial through field adapted parasites?
Sorry, can you repeat that?
Yeah, what's your take on in vitro evolution against these new antimalarials through field adapted parasites?
Oh yeah, I think that's another important thing that we need to look at. So everything that we've done has been primarily in a couple of different laboratory strains that were isolated, 30 or 40 years ago. And they don't have any of the background mutations that you would have in a modern artemisinin-resistant isolate. So, will we find the same things? Do we have the same drug resistance transporters coming up when we move fields to a different strain? And I think that's an experiment we haven't done yet, but one that we're hoping to do.
Okay. And is it general phenomena to mutate for parasite when it grows in sublethal concentration of any inhibitor? I mean, does it happen always?
Yeah, I think that you can, there are some compounds where it's very difficult to get resistance, and then there's some compounds where resistance comes very readily. For example, if you have something that's hitting cytochrome BC1, you often get mutations very quickly, but it's also true that these are mitochondrially encoded mutations, and you have many more copies of the mitochondria than you have for other chromosomes. And so you might expect that you have mutations in that more readily. Another thing that can contribute to the speed at which you get resistance has to do with the number of different ways to get resistance. So for some compounds, there's only one mutation that you can potentially get and then for other compounds, you can get an MDR1 mutation, you can get an amplification, and you can get multiple point mutations. There's a target called dihydroorotate dehydrogenase. We've studied that fairly extensively, and there's a lot of different ways that you can get mutations that give you resistance to dihydroorotate dehydrogenase. So I think there's probably a mathematical relationship between how easy it is to get resistance and the number of genome replications and the number of paths to resistance, and then ultimately you might be able to predict this, but we don't have quite enough data.
Okay, the final one, what is the source of these compounds? Are these chemically synthesized or nature derived? Also, was any natural compound was used in the screening?
Yeah, that's a very good question too. We have used both. So K609 came from a natural product library, but it was actually synthetic, that someone had put it in because it was natural product-like. Cladosporin is a compound we've worked on that's a natural product, and that inhibits lysyl tRNA synthetase. And that helped us identify lysyl tRNA synthetase as a target. But other compounds have been, are synthetic. So we'll take any some of the natural products have given us very spectacular results in terms of finding a target, like carmaphycin, which targets the proteasome, for example. But it's also true that some of the natural products, it's a little bit more difficult to get resistance. They're fairly large molecules. It may be that they interact with the protein at multiple spots.
Okay, so with this, I would like to close this session, and I know it's pretty late for you.
Oh,
so,
yeah, I can talk for a few more minutes if you want.
So I would like to thank you on behalf of NMR and MERA-India, and thank you very much for accepting our request. And I would also announce that in this series, in the next lecture would be from Professor Philip Rosenthal from University of California, San Francisco. And over to the director, NMR, to say a few words to Professor Elizabeth.
Thank you, Dr. Winzeler. That was a really enlightening sort of talk on all the products that potentially could be the next generation antimalarials. I just had a quick question. Sorry, I should have asked earlier in the chat box, but is the target of sulfadoxine drugs, this DHPS enzyme, is that being, even the whole pathway for that matter, are they being tracked? Because, the target is validated. The drugs worked for a while till resistance set in. But I mean, this approach of looking for new targets when the target is actually there, how about looking for other compounds which are not sulfa-based, for example? I'm just wondering.
That's valid. We don't, I mean, the approach I've described is one that's given us a lot of material to work on, but we aren't particularly exclusive, and there are some targets that we're considering that came from other methods, and DHPS is not one that's currently in our list, but I think could be one that we've actually discussed. And it's clinically validated, so it's a good target. We tend to stay away from like DHFR just because DHFR inhibitors have been used so extensively. There's a lot of background mutations, which make DHFR, for example, a possibly questionable target to pursue. But there are other ones that we are definitely considering. So it doesn't have to be discovered through in vitro evolution. If we can get a target from some other method, we will be, whether it's historical or for proteomics, we'll take that as well. There just haven't been many targets that have really been discovered using proteomic methods in my.
Yeah, no, that's fine. And I have another small question for you. Diane Wirth, there was a paper that about proline tRNA synthetase where it was understood at the time that perhaps there was an epigenetic phenomenon.
Yeah, I think we did some more sequencing on that, and I think we found that there was a transporter mutation that had been possibly overlooked.
Okay, got it. So then everything fits. Perfect. That's a happy ending there. Thank you from our side again, Dr. Winzeler.
I mean, don't quote me on that, but because I don't think it's published yet, but I think I'm not sure that she's sticking with that theory at this point, but it remains a possibility.
Sure, I understand. That's perfectly fine. Thank you again from all of us at NIMR.
Yeah, thank you.
Bye-bye.
Have a good evening.
All right, bye.